Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Nazatun Najihah, and today I'm going to be presenting the use of DNA technology in forensic dentistry. I'm going to start our discussion with the introduction of this research. This research provides a comprehensive review of the recent advancements in current concept in forensic DNA profiling, particularly emphasizing its application in forensic dentistry. It highlights the crucial role of teeth as resilient sources of high-quality DNA in the identification of individuals during mass disasters, criminal investigations, and paternity testing. The review discusses various DNA analysis techniques, including STRs, mtDNA, SNPs, and Y chromosome analysis, and evaluate their usefulness, advantages, and limitations in forensic contexts. This research contains several objectives, which include to give a thorough rundown on the most current ideas, developments, and patterns of forensic dentistry DNA fingerprinting, to emphasize the importance of teeth as a dependable supply of high-quality DNA, particularly under difficult situations where other tissues may be degraded, like mass disasters, trauma, or decomposition. Next, to highlight the advancements of technology from conventional DNA markers like VT-NRs to contemporary methods like SNP analysis and mtDNA, demonstrating its uses in sex, ethnicity, physical trait determination, and human identity. Last but not least, the study emphasizes how forensic DNA research is always changing and how important it is to improve the precision and dependability in forensic investigation.
So moving to the next, background on the role of DNA technology in forensic dentistry. Forensic dentistry plays a vital role in identifying individuals, especially in situations where the body is severely damaged, such as in situations of fires, decomposition, or explosion. In these cases, traditional identification methods like facial recognition or fingerprint are often not possible. Historically, identification relied mainly on blood group testing and serological analysis. However, this method has limited precision and couldn't always confirm someone's identity accurately.
The turning point came with two major scientific discoveries, which is first the discovery of DNA double helix structure by Watson and Crick in 1953, which revealed the foundation of genetic inheritance. Second, the development of DNA fingerprinting by Jeffrey and Kulik in 1985, which introduced a way to identify individuals based on their unique DNA sequences. Since then, DNA profiling has become widely accepted in courts and is now considered a reliable and powerful method for solving the criminal investigation, conducting the paternity tests, and identifying human remains that are decomposed or damaged. Forensic dentistry specifically benefits from these advancements by using both genomic and mitochondrial DNA extracted from the teeth. So teeth are often well preserved even under extreme conditions, making them especially valuable even when other body parts are no longer usable for identification.
So now we move to the next part, which is the problem statement of this research. In forensic cases, identifying human remains can be very difficult, especially when there is very little biological material left to examine. This is because the human body decomposes very quickly after death, especially in extreme situations like fires, explosions, or natural disasters. However, teeth are one of the most durable parts of the human body. They can survive extreme conditions and are often the only part left that still contains usable DNA. Next, normally, identification is done using fingerprints, blood, hair, or saliva, or any other body fluid. But in disaster cases, these materials are often destroyed or unavailable. So we need a more reliable source of evidence, and DNA from teeth can be that solution, as it offers better chances of accurate identification.
Now, I would like to talk about the significance of the study. Forensic dentistry plays a significant role in identifying victims of major disasters and resolving complex criminal cases. In recent years, the world has seen numerous mass disasters, including bombing, earthquake, storm, tsunamis, plane crashes, railroad accidents, and terrorist attacks. In this situation, it becomes challenging to identify a person precisely. In both natural and man-made crisis scenarios, dental identification has always been crucial, since teeth and jaws can withstand extremely high or low temperatures and offer important hints for identifying a person. The dental records such as casts, models, radiographs will be very valuable for comparing with post-mortem records in case of mass casualties. Therefore, the use of DNA technology in dentistry is essential to perform identification.
How we prepare the sample for forensic DNA analysis and the key application of DNA in forensic dentistry. Firstly, the sample we work with can include the various biological materials such as tooth pulp, dentin, cementum, blood, bones, hair, or even artifacts like chewing gum or cigarettes. And these materials are valuable sources of DNA, especially when other traditional methods are not possible. To extract the DNA, we follow several essential steps, which is first, we break open the cells in the sample, and this process is called lysis, which is to release the DNA. Next, we remove the proteins and other contaminants to ensure we get the pure DNA. Then we isolate and purify the DNA, often using the special techniques. There are different methods for DNA extraction, such as the phenol chlorophyll method, which uses organic solvents, the Shellax resin method, which is quick and effective, and the FTA pepper technology, which simplify the collection and storage, and alcohol-based method like using the isopropanol or ethanol for DNA precipitation. Choosing the right method depends on the quality and condition of the sample. For example, highly degraded or burnt sample might need more specialized techniques to recover usable DNA. Once we have the DNA, proper handling is crucial to avoid contamination. Samples should be stored properly, either dry or frozen, and protected from the heat, humidity, or chemical exposure that could damage the DNA. In forensic cases, this step ensures we retrieve reliable DNA profiles. Different methods may be required for tough or compromised samples, such as those exposed to extreme conditions. Next, let me walk you through some of the key ways DNA is applied in forensic dentistry today. First and foremost, human identification is a major application. DNA extracted from DNA tissues like a pulp, dentin, or a cementum can definitely confirm a person's identity, especially in challenging situations like the severe burns or decomposition where traditional methods might not work. Another important use is the analysis of bite mark evidence. Saliva left at the bite mark site can be sampled to extract DNA, which can help to identify or eliminate suspects involved in assault or abuse cases. In mass disasters such as plane crashes or natural calamities, DNA from teeth or bones become critical. Because dental tissues, especially enamel and pulp, are resistant to environmental damage, they can provide usable DNA even after extreme conditions like incineration. Sex determination is another application. By analyzing the DNA from dental tissues, forensic experts can identify an individual's gender using chromosomal markers, even from heavily burnt or degraded samples. When dealing with degraded samples, for example Asian remains or severely burned bodies, specialized extraction methods are employed to recover the viable DNA. Sometimes, if nuclear DNA isn't available, scientists turn to mitochondrial DNA, which is more abundant and can be more informative for degraded tissues. Legal and criminal justice also heavily rely on DNA analysis. Many countries, including Brazil, have laws approving the use of DNA profiling as evidence in court, which has strengthened the reliability and credibility of forensic investigations. To facilitate ongoing investigation, DNA banks are being established. Lastly, DNA can be extracted from various artifacts involved in crimes like chewing gums, cigarettes, or even bite marks in food, adding another dimension to evidence collection. These diverse applications demonstrate how integral DNA analysis has become to forensic dentistry, making the identification faster, more accurate, and possible even in the most challenging cases.
So now I will explain the DNA extraction method that are commonly used in forensic dentistry, especially when working with teeth samples for human identification. So the first method is the organic method, which uses phenol chloroform to break open cells and separate DNA from other materials like proteins. This method is mainly used when we want to extract high molecular weight genomic DNA, which means long and intact DNA strands that give more accurate results in testing. So the main advantages of the organic method is that it produces high quality DNA, which is very useful for forensic identification. However, the disadvantages are that it is time consuming and involves several steps, which increases the risk of contamination if not done carefully. The second method is the Shellax 100 method, which uses a chelating resin. This resin binds to metal ions that can damage the DNA and helps protect the DNA during extraction. So the biggest advantage of this method is that it is faster than the organic method and has a lower risk of contamination. So therefore, this makes it very useful when working with small or slightly degraded samples like those often found in forensic cases. But the downside is that it is expensive and not every lab may have access to this method. The third method is the FTA paper method. This is used mostly for quick storage and processing of DNA. It is very convenient and easy to use, where you just apply the sample onto the paper and it stores the DNA safely for later testing. However, it is also expensive and less commonly used in forensic labs compared to the other two methods.
In this research paper, the researchers analyzed three main ideas. The first one is the effectiveness of DNA extraction methods. The second one is types of DNA and their forensic values. And lastly, the use of PCR and STR in identification. For the effectiveness of DNA extraction methods, the researchers analyzed several DNA extraction techniques, which includes organic method, Silex 100 and FTA paper. The second analysis is types of DNA and their forensic value. There are two key types of DNA for identification. The first one is genomic DNA. It is found in the nucleus of each cell and provides genetic information. And the second one is mitochondrial DNA, which is usually found in mitochondria. It is another type of material that can be used for body identification. It is useful for degraded and small samples. When extracted the DNA, the likelihood of obtaining DNA from mitochondrial DNA is higher than the found in genomic DNA. It can help narrow down identity in missing person or disaster victim character. The last research is the use of PCR and STR in application. So how does PCR and STR helps in identification? PCR is a polymerase chain reaction. It can produce millions of copies of specific DNA regions from tiny biological samples, while STR is short tandem repeats. It is short sequences of DNA repeated in tandem at specific locations in the genome. STR profiling is used to create a DNA profile from the tooth. The profile is compared with antemortem DNA from the person, relatives, and DNA databases.
Now, I will explain the recommendations and final plan for implementation based on the findings from this research. First, it is recommended to use teeth as a reliable source of DNA, as teeth are known to be strong and durable even in extreme conditions such as fire, decomposition, or trauma. Because inside the tooth, especially in the dental pulp, there is a rich supply of high-quality DNA, making it an excellent sample for forensic identification. Because of this, teeth should be prioritized as a DNA source in challenging forensic cases. The second recommendation is to standardize the DNA extraction protocols. Currently, there are several different methods used in different laboratories, but no single universal protocol. So by creating a standard procedure for extracting DNA from teeth, it ensures that the results are more consistent, accurate, and legally acceptable. So this would also help classify DNA from teeth as reliable forensic evidence at a crime scene. Lastly, it is important to expand and support public DNA testing facilities. Many regions still lack access to proper forensic DNA testing, especially for dental samples. So the government and related institutions should invest in setting up more public DNA labs so that DNA testing can be done more quickly and affordably. This is especially important in cases involving burned, decomposed, or unidentified bodies, where DNA from teeth may be the only option for the identification of humans.
To support the findings of this research, several references had been used, which extracted from findings by Alvarez et al., Shiroma et al., and Marjanovic et al. They mentioned that teeth are a great source of DNA analysis in forensic investigations, since they are resistant to harmful conditions like decomposition, trauma, mutilation, as well as incineration. Also, a tooth can yield a large amount of DNA, a high quality, which is a significant benefit for DNA research. Not forget to mention, due to DNA profiling, the technology became more sensitive, reproducible, suitable to computer databases. It quickly became the industry standard forensic DNA system used for paternity testing and criminal casework. Smith et al. Mentioned that dental pulp can be extracted from pulp chamber using standard excess cavity preparation because the pulp is also a rich source of genetic material. The method's benefits include simplicity, affordability, and preservation of tooth integrity, all of which can be taken into account in forensic examinations.
Therefore, from this research paper, it can be concluded that teeth are a robust and excellent source of DNA, which are useful in forensic investigation. DNA technology, especially PCR and STR analysis, enables accurate identification even from small or degraded biological samples. Therefore, forensic dentists must be trained and equipped to use modern DNA methods, as their role is increasingly crucial in legal and humanitarian identification work.